Now, our discussing topic double beam spectrophotometer or spectroscopy presented by P. Trinadh, lecturer in biotechnology, skbr College, Amalapuram. Sem 3, Unit 1. Our topic double beam spectrophotometry. In the introduction of double beam spectrophotometry, the purpose of this instrument is to detect the amount of light of a specific wavelength observed by an analyte in a sample. The double beam spectrophotometry has two light paths. Both are originating for the sample light source. The two light paths are one path is for the sample and other path for blank or the reference. In this simple diagram, a schematic representation on the double beam spectrum, you observe the wave light sources. First light source, diffraction grafting, slit, rotating disc, sample cell, mirror, reference cell, detect and computer, and chart recorder. First on light source. Light source generally using sodium lamp or deuterium lamp for UV and tungsten for double beam spectrum. Slit. These slits are narrow flow the light source. Rotating discs. These rotating discs are two in double beam spectrum, one for UV and another for double beam spectrum. Actually double beam spectrum means a single light split into two parts and give to the result. The two parts of this splitting light rays is known as double beam spectrum. In these between, mirror has on sample cell the light passing light source through a sample slit and rotating disc into passing through the mirror. Those mirror are reflected on one for rotating disc and another side a rotating disc or mirror to reference cell. One light path from sample cell and another from rotating cell. These two are connected and give to the detecting and the computer and also represented by chartly recorded. Light source. The double beam spectrum for the light source is split into two. One beam is illuminate reference standard and the other illuminate the sample. The light source which give the entire visible spectrum. The narrow ultraviolet so that you are covering the range for about 200 nanometers to about 800 nanometers. In this diagram, the light source is split into two parts by using slit. This slit gives to the narrow flow of the light. Its range 200 nanometers to 800 nanometers near the ultraviolet region. Diffraction. Diffraction grafting is an optical compound with a periodic structure which split and diffract into the several beam traveling in different directions. The slit only allow light of a narrow range wavelength through into the rest of the spectrometer. Rotating disc. In these double beam spectrum mainly based on to reflect by using rotating discs. The rotating discs are three sections. The rotating disc have three sections: transparent section, mirrored section, and blank section. The light coming for the diffraction grafting and slit will hit the rotating disc, and one of three things can happen. That means In this diagram, when a light source, blue color lined, hit on the slit, what will happen? It turns into a three sections. That means one is sample cell and another one travel from sample cell by using rotating disc, and other one travels from reference cells by using mirror arranged on the downwards of the rotating disc, and these two light rays are immerged or junctioned at the second rotating disc, and these give to the detecting and the component of the computer. So, if the light hits the transparent section, it will go straight through the passing through a cell containing the sample. So, it is then bounded by a mirror onto a second rotating disc. The disc is rotating such that when the light arrives from the first disc, it meets the mirrored section of the second disc. The bounce it onto the detector. That means in the following diagram, you observe the transparent light hit on the first mirror or rotating disc. So these into in the sample cell, and the mirror is reflected into the mirroid region. So these mirroid region give to the detecting. The sample cell containing a solution, those to detected by the mirroid region or second rotating disc. If the original beam of the light for the slit hits the mirroid section of the first rotating disc, it is bounced down along the pathway. After the mirror, it passes through the reference cell. Finally, the light gets to the second disc, which is rotating in such a way that it meets the transparent section. It goes straight to the detectors. In this diagram, the vertical beam of light hit in mirroid region and passing downwards to mirror and reflected into a reference cell and transplanted to give the mirroid region. So this transplanted section gives to the sample to detect the detector region, which are maintained by computer. If the light meets the first disc at the black section, it is blanked, and for a very short will no light pass through the spectrophotometer. These two measure a amount of light produced by the detector. The sample and reference cell. Generally, the sample holder or sample cell, which used to be made up of glass or quartz containers. They are often designed so that the light beam travels a distance of 1 centimeter through the contains. The sample can be gases or liquids. Maybe in double beam spectrum mostly used liquids. UV also using liquids to quantitative or qualitative analysis. The sample cell contains a solution of the substance. You are tested usually very diluted with solvent. The reference cell just contains the pure solvent. The detector and computer. The detector converts the incoming light into a current. The higher the current, the greater the intensity of the light. For each wavelength of light passes through the spectrometer, the intensity of light passes through the reference cell is measured. This is usually referred to as I naught. That I for intensity. The intensity of light passes through the sample cell is also measured for that wavelength. In this formula, A equal to log 10 I naught by I, where the sample bit of maths is then done in computer to convert into something called the absorbance of the sample. Give the symbol A. The relationship between A and the two intensities is given by formula A equal to log 10 I naught by I. These formula also derived from Bayer-Lambert's law. We also studied on UV spectrum. And finally, a double beam spectrum to gives the graphical representation to the detector, and also computerized chart. Advantages of double beam spectrum are that light source variations are composite has a sensitive changes in the detector. That means the light source mainly filterized by sample solution or rises make more stable. It gives to the stable result. The double beam instrument allows you measure the sample and reference standard at the same time. The improved analytical precision and sensitivity also observed by using double beam spectrum. Speed of operation. This is also accuracy short time to detect the analysis the sample. Disadvantages. The only one disadvantage is it is costly. It is instrument is very cost. Thanks to all. Presented by P. Trinadh.